oligosaccharide, it becomes a polysaccharide. Most carbohydrates found in nature occur as polysaccharides. Polysaccharides are also known as glycans. To simplify things, polysaccharides can be a homopolysaccharide or they can be a heteropolysaccharide. A homopolysaccharide means the polysaccharide only contains a single type of monosaccharide. For example, it only contains glucose molecules linked together. A heteropolysaccharide means that the polysaccharide contains two or more different monosaccharides. So for example, a long chain of fructose and glucose molecules. To make things a little bit more interesting, a polysaccharide can also be unbranched, like what you see here, or it can be branched. This goes for both homopolysaccharides as well as heteropolysaccharides. Heteropolysaccharides can also be unbranched or branched. What you have to understand is that polysaccharides I am currently drawing are very small. In reality, the polysaccharide contains thousands, are made up of thousands of monosaccharides linked together. In this section of the video, we won't focus on heteropolysaccharides, but we will look at homopolysaccharides. Because homopolysaccharides serve as storage forms of monosaccharides in both humans and plants, and even bacteria. So they're very, very important. So let's look at some examples of homopolysaccharides. Starch is a storage form of monosaccharides in plants. Starch is the main carbohydrate in the human diet and are found in our bread, cereal, and rice. Starch is only made up of glucose because it is a homopolysaccharide. Let us zoom into this area here and learn a bit about the bonds. So here we have our regular alpha 1,2,4 glycosidic bond between two glucose molecules. And this is because carbon number 1 and carbon number 4 of these glucose molecules are involved in the linking process. However, the branching points here is actually an alpha 1,2,6 glycosidic bond between two glucose molecules. And that is because carbon number 1 of this glucose and carbon number 6 here are involved in the linking process. So what to take out of this is that starch has two forms. It can be branched, like what I just explained, or starch can be unbranched. If starch is unbranched, so it is only a chain of glucose linked together by alpha 1,2,4 glycosidic bonds, it is referred to as amylose. If starch is branched, it contains both alpha 1,2,4 and alpha 1,2,6 glycosidic bonds between glucose, and therefore it is referred to as amylopectin. Amylose and amylopectin are two forms of glucose polymers. The other good example of a polysaccharide is glycogen. Now, glycogen is a homopolysaccharide because it is made up of glucose. Glycogen can also be branched or unbranched. Glycogen is a storage form of glucose in animals, such as humans. Starch and glycogen are actually very similar in structure. They both are made up of glucose, and they can either be branched or unbranched. So both starch and glycogen contain amylose and amylopectin. The only difference is that glycogen has these branch points occurring every 8 to 12 glucose residues. In starch, these branch points occur every 24 to 30 glucose residues. So the branch points occur more frequently in glycogen than starch. And this, of course, will influence the structure in some way. Another type of polysaccharide is dextrans. Dextrans are structural components in bacteria and yeast. These polysaccharides are made up of alpha 1,2,3 and alpha 1,2,6 glycosidic bonds. So here we have glucose units with alpha 1,2,3 bonds and alpha 1,2,6 bonds. However, the dextrans can also contain alpha 1 to 2 and alpha 1 to 4 glycosidic bonds. Finally, the other polysaccharide worth mentioning is cellulose. Cellulose are structural components in plants. They make up the plant cell wall. They are unbranched homopolysaccharide consisting of thousands of glucose molecules. So here you can see unbranched cellulose on top of each other. In cellulose, the glucose molecules have a beta configuration. And therefore, the bonds between these glucose molecules are beta bonds. The glucose molecules are linked together by beta 1 to 4 glycosidic bonds. Humans do not have enzymes that break down, that hydrolyze beta 1 to 4 glycosidic bonds of cellulose. And so humans cannot digest cellulose. Now, even though cellulose are only chains of beta glucose, so unbranched beta glucose, these chains can form hydrogen bonds with each other, forming a very strong structure.